Hello, welcome this afternoon. My name is Barbara Stone, and I will be talking to you today about the future of mouse embryo transfer, achieving the 3 R's with the NSET device. During the talk, if you have any questions that you would like to have us address, please feel free to log a question, and we will get to the question and answer session at the end of the webinar. First, I'd like to talk to you a little bit about what is embryo transfer. Quite simply, it's the removal of embryos from a donor animal and placement of those embryos into a recipient. Both surgical and non-surgical methods are available, and the use of embryo transfer allows researchers to perform manipulations in embryos for research. And these manipulations occur in vitro. Embryo manipulations in mice are used for generating genetically modified mice. They're used for rederivation or removal of pathogens from mice, for breeding purposes, and also for recovery of strains, including and after disasters or for storage purposes. Some common embryo procedures include DNA microinjection, cryopreservation of embryos, in vitro fertilization, and also embryonic stem cell-based transgenesis. Regardless of the techniques that are used, embryos must be transferred to a suitable recipient for development. Traditional mouse embryo transfer methods have been surgical, so why exactly is there a need for non-surgical methods? And as you probably know, Russell and Birch defined the 3 R's in their book, The Principles of Humane Experimental Technique. The 3 R's are an ethical framework for animal use in scientific experimentation. They include replacement of animal models, reduction in the number of animals used, and refinement of techniques, for example, reducing pain and improving animal welfare. An example of refinement in a technique is the non-surgical embryo transfer procedure. There are several methods for embryo transfer in mice. Oviduct surgery is used for early stage embryos. Uterine surgery is used for morula and blastocyst stage embryos, and non-surgical embryo transfer can also be used for morula and blastocyst stage embryos. Non-surgical embryo transfer with the NSET device is a 3 R's refinement technique that reduces stress in mice. What I'd like to show you here is that the NSET procedure is effective for embryo transfer of blastocysts. And blastocysts were transferred to the uterine horn of 2.5 days postcoitum or DPC pseudopregnant female CD1 mice. The pregnancy rate shown here is the percent of females becoming pregnant after embryo transfer, and the birth rate is the percent of embryos transferred to females that become pregnant. And for the non-surgical embryo transfer procedure, you can see that the pregnancy rate is approximately 82%, and the birth rate is 46%. So while non-surgical embryo transfer is effective for embryo transfer, the question remains, does it meet the criteria of a 3 R's refinement? So in order to determine this, first we tested induction of stress hormones during surgical and non-surgical embryo transfer. And we were able to determine that stress hormone levels are higher in response to surgery than non-surgical transfer. Now, for example, if you have a stressor such as pain, it will signal the hypothalamus to make corticotropin-releasing hormone, which then begins a cascade, resulting in the release of corticosterone. This cascade can affect heart rate, lung function, and other things, and generates an overall stress response. Corticosterone levels can be measured either in the blood or in the feces, and here we chose to measure fecal corticosterone levels. And this is a fold increase in corticosterone levels here on the left. Fecal samples were collected from pseudopregnant female mice at 2.5 days postcoitum and assigned to 5 experimental groups. The first experimental group is a control group, and then a non-surgical embryo transfer group, a surgical transfer group, also as controls, non-surgical embryo transfer performed with anesthesia and an anesthesia control. Corticosterone levels in fecal samples were measured by ELISA assay. And as you can see indicated by the red bars, the corticosterone levels were significantly increased in mice 3 to 6 hours post-surgical transfer, but there was no significant increase for non-surgical embryo transfer or for any of the controls. Next, we tested cardiac responses to the surgical and non-surgical procedures. We used electrocardiograms, and electrocardiograms are recordings of the electrical currents that pass through the heart and signify the activity associated with each heartbeat. The non-invasive EC-Genie instrument for mouse specifics was used to record these electrocardiograms from mice without restraint or without any surgical implants. Readings were made by placing the mouse on a sensory pad outfitted with 3 monitoring electrodes. The cardiac activity was measured passively through the pause. In this experiment, heartbeats per minute are graphed at various hours post-procedure for, again, the 2.5 DPC pseudopregnant CD1 mice. Data was analyzed by mouse specifics using EZCG analysis software. And what you can see here is actually an anesthesia effect, in that the mice that were either had surgery performed on them or were anesthesia controls or NSET with anesthesia controls all had a decrease in the number of heartbeats per minute by 38 to 50%, whereas the NSET procedure and the no procedure control did not alter the heartbeats per minute. Variability in beat-to-beat rhythm of the heart is measured as a coefficient of variance, and here we have the coefficient of variance graphed at various hours post-procedure, again for the pseudopregnant CD1 mice. And what this shows is that variability in cardiac rhythm is significantly increased by 8% for the surgical group, but not for any of the other groups that were tested. So in conclusion, the NSET procedure is effective for uterine transfer of blastocyst stage embryos. The surgery causes a significant increase in the levels of the stress hormone corticosterone, whereas the NSET procedure does not. Anesthesia also causes a 38 to 50% decrease in the number of heartbeats per minute, whereas the NSET procedure does not alter heartbeats per minute. And last, the surgery increases cardiac variability by 8%, whereas anesthesia alone and the NSET procedure do not significantly increase cardiac variability. Use of the NSET device is therefore less stressful to the mice than a surgical embryo transfer. And as such, it is a successful alternative to a surgical procedure and a 3 hours refinement for a currently used technique. So what are some of the applications to embryo transfer that non-surgical embryo transfer is applicable for? We've tested non-surgical embryo transfer with embryos that have been manipulated in various ways prior to transfer. And we have shown that NSET can be used to transfer microinjected embryos. Now these embryos are, this graph is a comparison of the transfer efficiency between embryos transferred by surgical and non-surgical methods after injection of a variety of different transgenes. In this case, 5 different transgenes were used. For surgical transfer, the microinjected embryos were transferred to the oviduct during oviduct surgery into 0.5 DPC pseudopregnant female mice. For non-surgical transfer, however, the microinjected embryos needed to be cultured to blastocyst stage prior to transfer. And these were then transferred to the uterine horn into 2.5-day post-coital pseudopregnant recipients. The transfer efficiency, as you can see, is similar between the two methods, and the number of transgenic pups recovered after the procedure is also very similar. Non-surgical embryo transfer can also be used to transfer embryos generated by in vitro fertilization. This graph shows that the pregnancy rate and the birth rate for non-surgical embryo transfer. IVF of fresh oocytes from B6C3F1 female mice and cryopreserved sperm from B6C3F1 males was performed. Fertilized embryos were then cultured in KSOM media to blastocyst stage and transferred into 2.5-day pseudopregnant females. Transfer of the IVF-derived blastocyst resulted in about a 60% pregnancy rate and a 43% birth rate for IVF. NSET can also be used to transfer cryopreserved embryos. Here, cryopreserved B6C3F1 one-cell embryos were cultured to blastocysts in KSI media and transferred again to 2.5-day pseudopregnant CD1 recipients. The transfer of the cryopreserved embryos resulted in a 50% pregnancy rate and a 39% birth rate for this experiment. Non-surgical embryo transfer can also be used to transfer embryos after ES cell injections. This data is courtesy of the microinjection service at the Jackson Laboratories. Here, C57 Black 6 blastocysts were injected with JM8A3N1 ES cells, and then they were transferred into the 2.5-day pseudopregnant CBY-B6F1J recipients. In this experiment, the pregnancy rate from NSET transfers of ES cell injected blastocysts the pregnancy rate was 80%. The birth rate was 31%, and the total rate of chimerism of number of chimeras per the number of pups surviving to weaning was 54%. We've also been able to successfully transfer sperm for artificial insemination with the NSET device. And so here I'm showing the pregnancy rate for this experiment was roughly 36%. And the picture on the right is a picture of one of the resulting pups from one of these experiments. In summary, the artificial insemination experiment actually was performed with superovulated females, and the sperm delivery occurred on the day after the second hormone injection. And I'm not going to go over the specifics of this particular experiment right now, but I will be happy to cover that later if anybody's interested. In conclusion, the NSET device can successfully transfer embryos after microinjection of DNA when cultured to blastocyst stage. It can successfully transfer blastocysts derived from in vitro fertilization. Also, blastocysts developed from cryopreserved early stage embryos. It can also transfer ES cell injected blastocysts, resulting in recovery of chimeric pups. And it can also be used to transfer sperm during artificial insemination, resulting in live births. A couple of other uses, which we're not going to be talking about today, but have been referenced in the literature, is that you can also transfer pathogens using the non-surgical embryo transfer device, and other materials, for example, Con-A-coated cephalose beads. Drugs can be delivered this way, and a multitude of other materials that can be used for research. This is a picture of the NSET device, and the NSET device is a Teflon catheter attached to a hub, which then attaches to a P2 pipette, which is used then for delivery of specified volumes of liquid. And as you can see here, it is individually packaged and ethylene oxide treated so that it can be used in sterile environments. And this device actually also comes with 2 specula in the ethylene oxide treated package, which the use of those specula we'll be talking about in a minute. The NSET procedure and device that I'm about to describe in detail was invented by Dr. Brett Spear and Michael Green at the University of Kentucky Transgenic Core Facility. They have described this in the literature, and there are 3 very simple steps to this procedure. First of all, load the embryos into the device, prepare the recipient mouse, and deliver the embryos. To prepare the embryos, first, the NSET device is attached to a P2 pipette. The embryos can then be loaded into the pipette while watching under a microscope. Up to 20 embryos can be loaded into the NSET device in 1.8 microliter of media. The volume is then adjusted to 2 microliters to introduce a small bubble into the bottom of the tip, which helps hold the liquid into the device. Next, the recipient mouse is prepared. A suitable female mouse is selected, and she needs to be 2.5 DPC pseudopregnant. The mouse is then held into position on the cage top. A small speculum is inserted into the vagina and then removed again, and then a larger speculum is inserted into the vagina. At that point, you can visually locate the cervix, which there's a picture of the way that the mouse is being held here, shown here. And on the next slide, you can see a close-up of a CD1 mouse with the larger of the 2 NSET specula inserted. And you can, hopefully you can see the cervix in there, and there's a small depression in the center, and that is what you would aim the NSET device at. Next, a video presentation of the procedure will be.
I've placed a 15 microliter drop of media under the lid of this petri dish, and I've loaded my embryos into it. Now I'm taking the NSET device, and I've set my P2 pipettement to 1.8 microliters. You simply place the NSET device onto the pipettement, look into your microscope, bring the tip down, and slowly release the plunger to load the embryos. Once you've loaded the NSET, then just dial up to 2.00 microliters on your pipettement. This will introduce a small air bubble at the tip, and that'll help keep your embryos from wicking out when they touch the inside of the mouse until you get it to where you're ready to expel the embryos. So the smaller piece of tubing is used to open up the vagina. Just insert it briefly, pull it back out. Now we're ready to insert the speculum. Just insert the speculum, adjust your lighting so that you can see down, and you can actually see the cervical opening. Pick up your NSET device, insert into the mouse until the hub touches the speculum. Expel the embryos, remove the NSET device, remove the speculum, and the procedure is complete.
So you'd like to see the video demonstration again, it can be seen on the Paratex website. Also on the Paratex website are complete instructions, frequently asked questions, and helpful hints and tips. I can be contacted directly also by the email address on this slide. I'd like to take a moment to mention a few of the most important tips that we have. First of all, removing the NSET device from the uterine horn without releasing the pipette plunger is highly recommended. The NSET device itself was designed as a single-use device, and so repeated use will clog the NSET tip with tissue, which could lead to unfavorable results. As always, inspection of the tip is good practice, just to make sure that the tip is not bent when inserted into the mouse, and also that any material hasn't clogged the tip. Please do not use lubricants during this procedure. They can clog the tip as well, and they are unneeded for insertion of the specula. And please make sure to read the NSET instruction sheet thoroughly before use. One of the most often asked questions is, which recipient strain should I use? And so we have compared a few different strains that are used for recipients as during non-surgical embryo transfer. And this graph shows strain-dependent pregnancy rates for embryo transfer methods. The pregnancy rate is shown on the left, and the various different strains are indicated below. Shown here are CD1 mice, Swiss Webster, B6 CBA F1s, C3Hs, and C57 Black 6. Also, we've tested B6 D2 F1 recipients, although not shown on this graph. I will include their results as well. What you can see is a comparison between non-surgical embryo transfer and surgery for each of these strains. If you look at the highest pregnancy rates here, you can see that the CD1 females, the B6 CBA F1 females, and the C3H females all have a very high rate of pregnancy. The B6D2 F1s also had an 80% pregnancy rate, and so they would be included in the better group of embryo recipients. On the next slide, you can see strain-dependent birth rates for embryo transfer methods. And here the birth rate's indicated on the left, the strain is indicated on the bottom, and there's a comparison between non-surgical embryo transfer and surgery. And what you can see here is that the CD1 females had the highest birth rate shown at 46%. The B6 D2 F1s also had a 44% birth rate, and so we're also very high. So what we've concluded from this is that many different strains can actually be used as recipients for embryo transfer. Strains in the labs are generally chosen for convenience and ease of use. A lot of people will try to use the strains that they already have, and we've shown that it's possible to do so. We recommend the 2.5 DPC CD1 pseudopregnant females for recipients here, mostly for three reasons. First of all, they have one of the highest pregnancy rates. Also, they have the highest birth rate, and they are extremely docile and easy to handle, and so as such, we prefer to use them here. Some of the other strains that have high birth rates and pregnancy rates tend to be a little more difficult to handle, and a lot of times aren't as good mothers, and so we prefer to use the CD1s. So some of the advantages of the NSET procedure itself. First and foremost, it eliminates a surgical procedure entirely. So there's no pain or distress to the animals. You can see from the video that the animal is very calm and easy to work with, and that animal had received no anesthesia or analgesia. Again, for the purposes of a non-surgical embryo transfer, there is no anesthesia required. There's no post-surgical monitoring. After the procedure is done, the animal is simply returned to the cage and the procedure is over. One of the most important things from a technical standpoint is that there's no surgical training required for this procedure. Learning the procedure is very easy. It's not something that requires a lot of technical training and can be very quickly implemented in your laboratory. The non-surgical embryo transfer procedure satisfies one of the 3 R's. It's a refinement of a technique. It reduces pain and distress. It's a terrific improvement for animal welfare. It also reduces regulatory burden. The need to explain why you need to use a surgery and how the surgery is going to be performed and all of the methods that are required to prepare your lab for a surgical procedure need not be performed. This is a non-invasive, non-surgical invasive procedure. It's extremely fast. The procedure itself only takes about a minute from start to finish, and that includes loading the embryos into the device, which is much faster than trying to perform a surgical procedure where the post-procedure monitoring even takes a large chunk of your research day. It's extremely convenient, and there's a significant cost savings involved, especially if you need to purchase surgical materials, an anesthesia machine, perhaps training technicians or researchers to do the procedures for a surgical procedure is very cost intensive, whereas the non-surgical embryo transfer procedure is much less expensive. So, in conclusion, I'd like to take a moment to acknowledge a few people and institutions that have helped with this research. First of all, the testing of the non-surgical embryo transfer device for ES cell-injected blastocysts was performed under contract at the Jackson Laboratory. We'd also like to thank Kim Carrico for her contributions towards developing the ELISA protocol, and Mouse Specifics for their generous support regarding the ECGene device and analysis of the data. This research is presented with support from the Office of the Director, National Institutes of Health, under the award numbers shown here, and by the Kentucky Cabinet for Economic Development, Office of Commercialization and Innovation, in conjunction with the Kentucky Science and Technology Corporation. I'd also like to thank our research team for all their hard work. Kendra Steele, James Hester, and Angelica Fath-Gooden were instrumental in performing these experiments and collecting the data. This is a list of selected NSET publications. I know that you can't read these here very well, but these references are also found on the Paratex website. If you have any interest in pathogen transfer or material transfer, those references are indicated on here, as well as the embryo transfer references. And I'd like to thank you all for joining me today. I'd be happy to take any questions at this time. Dr. Kendra Steele from the research team is joining me right now, and will be able to help answer any questions that have been submitted during the talk by the audience. And Kendra?
Thanks, Barb. Yeah, we have a few questions. I would like to ask that everyone keeps submitting those questions. The first one is, your artificial insemination data looks promising. Could you please share the details of that protocol?
Yeah, actually, let me get back to that slide. And, all right. So here you can see that the pregnancy rate for this artificial insemination experiment here was approximately 36%, I believe. And what was done here was, in general terms, we superovulated the female that was going to be the recipient of the sperm. And so the injections for superovulation are first to inject with pregnant mare serum gonadotropin. and then at a 47-hour interval, then injecting with human chorionic gonadotropin. Okay, and these were both done at levels which would normally be used for superovulation in a female, and so that was at 5 international units of both of those hormones. And those hormones were injected first at 8:30 p.m. for the first hormone and 8 p.m. at the second injection time point. And then the following morning, sperm were delivered using the NSET device from B6C3F1 males. Okay, so these males were euthanized in the morning, and the sperm was capacitated for an hour and a half. 40 microliters of the fresh sperm was transferred with the NSET device, and this occurred roughly at 9 a.m. on that morning. The females then were paired with vasectomized CD1 males overnight, and this is because the copulatory activity needs to occur in order for the pregnancy to be established. And the pregnancy rate that we then saw from that was 36%. And it was, I believe that we can actually improve upon this. We're working with the protocol right now to make it happen, but it was a very promising initial result.
Great. Okay. The next question is about the video. Is the mouse in the video anesthetized? And kind of a follow-up to that, aren't you afraid of biting?
The mouse in the video has not been anesthetized and is simply being held in a position which tends to make the mouse freeze in that position. And CD1 mice are very docile and easy to handle, and I've never been bitten by a CD1 mouse doing this procedure. Having said that, some of the other strains that we've compared the CD1 mouse to for pregnancy rate and embryo transfer efficiency, or birth rate, have been less happy about the procedure. And so we do have methods in place in our lab to work with less than cooperative mice, and this the only thing that we do is that we wear heavier gloves, and that seems to avoid any of the problems. But the CD1 mice are very calm and easy to use, and we've never had any problems with that.
Okay, good. How can I best be trained to use the NSET procedure?
Training for non-surgical embryo transfer, it can be minimal, or if you have a lot of experience with mice, or sometimes if you're uncomfortable with the procedure itself, it's helpful to take a class in this. And so there are two different things that we suggest. First of all, if you are an expert at handling animals and you're comfortable with the procedure itself, I would suggest either starting with euthanized animals, or you can also practice on animals that have been anesthetized. And if you are going to try something like that, make sure that the animal is indeed 2.5 days pseudopregnant. The opening of the cervix has to be dilated in order for the NSET device to pass through, and so if you try to do this without having the animal dilated first, it won't work. And so I would suggest definitely practicing, if you're new to this procedure, practicing a few times without embryos, because embryos are precious, and getting the hang of working with maybe euthanized mice first, and then moving on to animals that I don't think that the anesthetized mice are really necessary unless you're really uncomfortable with it. But moving on to animals that are just pseudopregnant in a live format, and you'll find that it's an extremely easy procedure to perform. If anybody is interested in being trained personally in this method, we do perform workshops every year internationally and locally, and we can also do workshops on site if anybody's interested in that. And so it's a very easy it's a very easy procedure to learn.
Okay. Do you offer free NSET samples?
We do actually offer samples, and I believe that we have a booth here in the BioConference Live, and you can actually request a sample at the booth, or you can request a sample from the Paratex website.
And that's all the questions we have for now.
Oh, all right. Well, thank you again, everybody, for joining us. And if you do have any further questions, we will be available on the booth in the booth area. And if you would like to contact us personally, feel free to do so. All of our contact information is on the Paratex website. And again, thank you very much for joining us.